I'm going to talk to you again about the point spread function. We're going to talk a lot about the point spread function during this this course and the connection with resolution to try to put the concepts that we have been seeing a little bit all together. So I remind you that the light that is emitted by the sample reaches part of this light because the sample emits in all directions, but only part of this light goes back into the objective. And when it reaches the edge of the objective, it diffracts because this is a hard boundary. And so the light bends around the edges of the objective. So the edges act like an aperture, and it's an iris outside of the image plane, and it affects the whole image. So this is what we have seen. If you have a field stop, you will see the edges like a curtain. The outside will become black when you close the field stop, but if it's an aperture, so the iris outside an image plane, then it affects the whole image. And this creates inside the at the back focal plane of the objective and at the final image plane, it creates a complex 3D interference pattern, which is what we image. So, and it's called the point spread function. So we have here our little fluorophore, or we have tons of them in our sample. And each of the fluorophores creates an image at the final image plane, which is bigger. So this is why it's called spread, right? It's a point spread. And because this behavior of light is very reproducible and very predictable, we can mathematically calculate it, and therefore it's called point spread function. Okay, so this is why it looks in XY, and it's bigger than the original dot, and in Z it's very elongated. And if you remember, we see some sort of a rugby man, and this is the image of every fluorophore in our sample is a complex 3D interference pattern called the point spread function. So here you can see the little rugby man, and then I was playing a little bit and going like, okay, it's like a rugby man with hula hoop rings. And so I used the AI to generate this. I thought it was, but it doesn't really look like our sample. But anyway, at least one of my attempts to generate rugby man with hula hoop rings worked. So what happens when there's more than one dot? Maybe we'll get a rugby crumb. Here we go. So how does the PSF relate to resolution when we have more than one dot? So we know that the resolution is basically the distance between two dots that we can identify as two dots and not just one. So they do not fuse. So this minimum distance, this is what the resolution is. So here we can see several dots, and here they're a little bit far away. So we can see two dots, then they get a bit closer to each other. And in here we have one dot diameter. So the distance between these two is just one dot diameter, and we can still resolve them. We can still say it's two of them. Then they get closer and closer and closer. And at some point here, we have only one radius away. So the two dots, if you want to say, they are like sort of overlapping and they're just one radius away. And now we cannot resolve them anymore. Okay. And if they get even closer, we really clearly see only one dot and it's not even elongated. So the resolution is the smallest distance that we can measure reliably. And the smallest distance, so a small distance means a high resolution. So high resolution, this is what everybody wants. It means a small number, okay? So all objects that are smaller, this is also something that it's important to think. All the objects that are smaller than the point spread function, or individual objects, just one object, it will look anyway like the point spread function. So you can imagine that our fluorophore in our sample is very tiny, but what we are saying when we're imaging is the point spread function. So even if the fluorophore is much, much, much smaller than the point spread function, the image cannot be smaller than the point spread function, okay? So any object that has the size of the point spread function or smaller will look like they have the same size. And so the smallest distance that we can resolve is the radius of the PSF. So all this can be calculated quite easily, and these are the equations that I gave you. So we can have a look a little bit at what these equations mean. I'm just going to pin myself, so click. There we go. Sorry, I go the wrong way. Here we go. So if you go online, I've said before, you can find all sorts of different resolution equations, and I'm going to tell you why now and why I've chosen those that I've chosen. So here we see the point sprite function in xy. It comes from this tiny dot, but it's much bigger. And here it is very elongated in z. And this can be represented also this way. We have the rings here, these rings, and then we have the bright center, which is here, and then more rings. So now these two dots are at a distance that is quite big. It's more than the radius. So the radius of the dot here, so the distance between these two dots is more than the radius of one dot. But now they are going closer to each other, and now we have only one radius between the dots, the center of the dots. And now this is smaller than one radius, so they overlap, and we are not being able to resolve them anymore. So the equation that we are using is the 0.61 lambda divided by numerical aperture. It's very common to use this equation for resolution in XY, and this is called the Rayleigh XY resolution. And it basically corresponds to the radius of the belly of the rugbyman. Okay, so this is what this calculates. And so it corresponds to this distance here. But this was an empirical measurement, as I said when someone asked. It was an empirical measurement that was done a long time ago. It's anyway used still, but the technique has evolved a lot. And at that time, there was no fluorescence. There's other equations that can be used. For example, the full width half max. So what is that? This is the maximum of the curve. And so if we go halfway, we have the half maximum. And the width here of the curve is the full width half maximum. And so this equation is 0.51 instead of 0.61. The rest is the same. So clearly this one will give you a better resolution. So it's more optimistic. But anyway, it doesn't matter because the curve is exactly the same. So at the end, you're measuring the same thing. It's just a different number that we're putting on that point spread function. So the full width half max is often used also to measure the fluorescence, the resolution. Then there is the Abbe equation, and the Abbe equation is even more optimistic because it's 0.5 instead of 0.51, and the rest is the same. But this is theoretical actually, and it is not possible to reach the Abbe limit completely because in some ways, we have this connection with the contrast, and at that resolution, we are actually losing the contrast, so we cannot see anything anymore. So it's just above. But so then you have equations that give you the excitation lambda instead, excitation wavelength instead of the emission wavelength. And that depends on if we use a confocal, a point confocal, or actually, yes, a point confocal or not a point confocal, then we will use either the excitation or the emission. But you can see that normally the excitation and emission curves, they're quite close to each other. So it doesn't make a huge difference if we use one or the other one. But of course, people who calculate exactly, I understand that they won't use the correct thing. But for us, maybe we don't care too much. And then, of course, there's a difference if we use confocal and wide field. So we should use the emission for wide field and the excitation for confocal. And then this is the Z resolution. So again, in here, we have the height of the blob. And this 2 lambda n divided by numerical aperture is this. So it is actually the radius of the whole thing, because this is actually it's a full blob, right? So this curve only shows half of it. So this is the radius, and this is what this equation calculates. So it's like the half of the rugbyman belly. And the real equation is something like this. If we have a pinhole that is 1 area unit or larger, and 1 area unit, as I've said before, is the size, it's the way we are setting up the pinhole of the confocal, and it changes based on the wavelength. So if we have a wider wavelength, we're going to have a wider area unit like this, but this is a way to sort of normalize it. 1 area unit just means from here to here, regardless of the wavelength. So you see the equation is quite complicated, but it's certainly more exact than what I use. But in any case, I've decided a long time ago that this is more than we actually need, because actually when you change equation, the numbers do not change so much in most of the case. And so in this course, we're going to use the X, Y, and Z resolution, which correspond to the radius of the point spread function in XY, so this guy, and the radius of the point spread function in Z, so this one. And we will do that for all systems. What the pinhole does when we are above 1 area unit, at 1 area unit or above, is that it increases the contrast. And we will see how there is a link between contrast and resolution in such a way that if we have no contrast, it doesn't matter if we have resolution. If we have no contrast, we cannot use this resolution. So there's this concept of usable resolution. Okay, so and then so this is what we're going to use, the radii, I think, of the main PFS, the PSF main spot. And yes, 1 area unit is used on point confocals to normalize the size of the pinhole for all the wavelengths. So you will get the same, I don't remember if I have something, that's it. Here we have 1 area unit, this is double of this Rayleigh resolution. And if the wavelength is bigger, it's going to go wider. But we can say 1 area unit for all the wavelengths. It just means that we go from the first 0 here to the next 0. So the resolution, how is the resolution linked to the sample design? So imagine that you want to count some large objects, for example, nuclei. They're quite big, right? But they are very crowded. So that happens in some tissues, they get compressed. For example, in the skin, it's sometimes difficult to count the nuclei, but you would like to count the number of cells, so you want to count the nuclei. If you label the nuclei with DAPI, this is what it will look like. And it's very difficult to resolve because what you need is a very high resolution where what you need is you need to resolve the distance between the two borders of the nuclei, even though they are very close to each other. So you need a really high resolution. Instead, you could label your nuclei this way, for example, with lamine, which is only in the nuclear membrane. And when you do that, even if your nuclei are very crowded, it will be much easier for you to quantify how many cells you have. And here you do not need such a high resolution. So because now what we have is the resolution we need is the distance from one border to another, not the distance here, but this distance. So we need a much, much lower resolution. We can get away with it. So you can see how there is a crosstalk between the resolution that is needed and required by the scientific question and the sample design. And it's really important to do this reverse thinking process where you start thinking image resolution and then you go backwards and design your sample and also your microscopy settings. So the design of the sample influences the resolution that is required to answer the scientific question. And the resolution is often not related to the size of the object, but to the distance between the objects. And in here, the distance that we need to resolve is the distance between two nuclei, whereas in here, the distance we need to resolve is actually from one side of the nucleus to another. And it's further away when the nuclei are crowded. Okay, so it's not so much about the size of the objects, but the distance between the objects, except if, for example, you want to, if your scientific question says that you want to measure the the size of an object. So for example, you want to measure the diameter of a mitochondrium. If this is what your scientific question is, then you need to have a resolution that will allow you to distinguish from one side to another of the mitochondrium, which is very small. So then you need an excellent resolution. So you have to think in terms of distance. What is it that I want to separate with my images? So it's the conclusion for this lecture is that the resolution is the smallest distance that we can measure reliably. And the objects that are smaller than the point spread function will appear to have the size of the point spread function. So you cannot have an image where the dots look smaller than the point spread function. And if you're imaging lines, for example, membranes or something like this, the thickness of the line cannot be thinner than the point spread function. So the point spread function has a huge impact. I mean, basically, this is what we're imaging, right? We're imaging the point spread function, so the impact is enormous. Anything that affects the point spread function will affect our image and our image analysis. And this is why we want to prepare our sample as best as possible to have no refraction, which lowers the contrast and means we cannot use the resolution that the objective is delivering. So it's not possible to resolve distances that are smaller than the point spread function radius in X, Y, or in Z. These are the two equations that we're using. And in this course, we will use for everything, all microscopes. I should have written camera-based and detector-based. We will use this equation, and this is the emission. And here we will use this equation for Z. So now, first, before I finish, we're going to do some exercises now, but I would like to know if there are some questions about this part.
Sylvie,
Yes?
the resolution that we counted gives the smallest distance that it can be measured between two objects.
Yes.
So the higher is this number, the better it is.
No, the opposite.
Oh, the opposite.
Because if the objects, so basically you want to separate objects.
Yes.
If they must be that far away for you to be able to separate them, it's not very good. But
Yes.
if they can be that close and you can still separate, it's very good.
Oh, yes.
So when we have a small resolution number, we have a high resolution. So maybe instead of saying high and low resolution, we should get used to saying good and bad.
Yes.
So small resolution number means a good resolution.
Yes.
Because then we can separate objects that are very close to each other.
Yeah, that's now clear. Yeah.
And you can see that, for example, if you have very tiny objects, right, and you want to count them, if they are not crowded, if they're far away from each other, it doesn't matter what size they have. You have no problem counting them. But if they are very crowded, they're very, very close to each other, and then the same object with the same size is very difficult to to resolve them and count them. So what we need to resolve is the distance between the objects. And if your question is to measure the size of an object, then the distance that you want to resolve is from one side of the object to another one. So then it's related to its size.
And it's the same with the Z resolution.
Same thing with Z.
Yeah.
Exactly. Which is a lot worse than XY.
Yeah. Thank you.
You're welcome.
Can I also ask something? So
Yes.
You said that we need a good resolution. For example, if you want to count objects like mitochondria, for example. What if my goal is not to count the objects, but the intensity rather? Is it still the same?
Exactly. Well, it depends how you formulate your question. And this is why it is so important to learn how to formulate your scientific question. If you're saying, I would like to know the intensity of each mitochondrion, then you have to separate them so that you identify each of them individually. If now you say, I would like to know the average intensity of the mitochondria in one cell, now what you need to separate is the whole cell. And then you measure the total intensity of the mitochondria inside the cell. So that is much easier. So we always want more. We always want to, get as much as possible. Well, we have to make do with the limitations of the macroscope. And it's much better to define a scientific question from the start that, allows you to, maybe you will take one more step and go to a high resolution, than designing your experiment from the start where it's actually impossible. And then you do a lot of work and then you have to go back. Or you try a lot and then maybe you get results that are not reliable. So when you define your scientific question, you need to figure out, what is it that I need to resolve in my image to answer this scientific question? Is this possible or not? If it's not possible, you have to change the scientific question or you change the technique.
Okay. Thanks.
So we will talk about the scientific question later. Have some more questions? I think Xu had a question.
Hi, can you hear me?
Yes.
Thank you. So I wonder, like when I count the RNA molecules, sometimes can happen the expression is really high. So I saw the pattern is really crowded. I wonder, is there any strategy to, I don't know, to make it quantification easier?
Question. Exactly, yeah. That's a good question, and we need to think that for RNA molecules, first we need to realize that we can also separate them with colors. So sometimes we have different type of species in the sample, and then we can have different colors, and therefore in each image, in each color, we have a longer distance between them.